get started. It is my great honor to introduce our second speaker, Dr. Emmanuel Candès. Dr. Candès holds the Garner-Simons Chair in Mathematics and Statistics at Stanford University, and is also a professor of electrical engineering. Over the span of the 15 years, from 2006 to 2021, he received 14 major awards, including the World Memorial Lectureship from INF and the MacArthur Genius Fellowship. He is also a member of both National Academy of Science and American Academy of Arts and Science. Dr. Candès' pioneering work, including compressed sensory, knockoffs, conformal prediction, a lot of them, has really profoundly shaped our field. So please join me, welcome Dr. Candès.
So I should say that membership to the Academy is more meaningful since Ken joined. I was not planning on saying anything about Ken. It's very fine. It's a good thing for us to be back with Kiara. Kiara is here. And we live not far away. And so I met Ken a long time ago when Kiara was in the genetics department. And from the very first day, I thought, this is the example of what a scholar should be. Like there was a high level of curiosity, some anecdotes, like I would go and get sandwich in the basement of Gonda. I don't know if it's still there. And I don't think there was a single lunch where I did not see Ken with a book reading about scholarly work of others. I think what I discovered through the years is also a very high level of scientific integrity and honesty, which really I thought I was very sensitive to. And the last thing I wanted to say was, a form of courtesy and kindness and the way he treats others, I think is really, in my view, a person we should all try to emulate. And I think the fact that we have this event year after year seems to be a testament of these incredible qualities, both as a scholar and as a human being. Okay, so I just wanted to talk to you a little bit about a very simple thing. My talk is not technical, and it's just about a little thing. And we're going to talk about model selective inference, and we're going to talk about drug discovery a lot. One thing I should dismiss immediately is that this picture was not generated by AI. It was generated by a fine artist whom I happen to like very much. And he has actually beautiful drawings about LA prior to this disastrous fires. I hope not too many of you are impacted by those. Okay. So this is joint work with Ying Jin, who is a former student of mine at Stanford, who is now at Harvard and will join Wharton. And then some of it is work with Yuri Leskovich, with this Stanford CS professor. And we have started a significant collaboration with Genentech. So I'll touch a bit on that. So I don't think I need to tell you that drug discovery takes time and is expensive. And people are trying to accelerate the process whereby we bring drugs to market. Of course, we have to examine a huge library of potential drugs. And so people are trying to accelerate this process. So, AI is all the rage at the moment. So, basically, the promise of the field is that we're going to move from in vivo experiment to in vitro experiment and now to in silico experiment. We're going to use the power of neural nets to actually select which drugs we should launch. And so there's a lot of popular articles about how AI or generative AI is going to change science and especially drug discovery, right? So the future of biotech is AI-driven discovery, nothing less. Okay, so this is a promise that now, because we have this kind of extraordinarily powerful predictive models, and the path to discovery is going to be significantly shorter and less expensive. Okay, and I don't know why is this working? Okay, so what we want to talk about today is that I have a library of compounds And I want to know which drugs are sufficiently active. So there's a disease of interest, for example, it might be HIV. The HIV has a target, which might be the reverse transcriptase that I want to block so that I'm not, the reverse transcriptase is I let it do its job, it's going to synthesize DNA that's going to be put in the host cell, and that's not good. And so I want to block this process. And so we're looking for drugs that would actually bind to the transcriptase so that you stop DNA synthesis eventually. And so now we're looking at enormous libraries of compounds with which could bind to the target. And so, the question is which drugs are sufficiently active? And so to do this, traditionally, we're going to rely on experiments, clinical trials, lab measurements, and so on and so forth. But of course, now we want to bypass this process. So traditionally, what we're doing is we go to the lab, we make measurements, and we measure the affinity of a possible compound to a target. Right? So I'm going to talk in this lecture about large outcome, meaning that I'm interested in drugs with a high affinity for a compound. And now, we say, well, that's too expensive. It requires a lot of money and time to actually make thousands and thousands of experiments. We'll see that people are sifting through catalogs of drugs that are in the hundreds of millions. So there's no way we're going to do that. And so we want to use AI-assisted approach to predict what I'm going to see in the lab, using a lot of new technologies such as AlphaFold and other things. Okay. So I want to use AI in lieu of real physical measurements so that I prioritize the drugs that I should study first and maybe send for clinical trials and stuff like that. Okay, so the problem setup is the following. We're going to assume that, I don't know if is this actually showing my slides? Is that showing my slides? It seems like I'm missing a lot of slides. Okay, anyway. Okay, so we're going to be interested in the following problem. So we're going to train a prediction model, mu hat, which takes the features of a compound, of a potential drug. This is very high dimensional for us. And it's going to map the features into what it thinks is a binding affinity. It can be a continuous variable in the case where I'm just trying to measure the affinity of a drug for a target, or it can be a binary, yes, it binds, no, it doesn't. And so X is very high dimensional. It's kind of whatever I know about my drug. For example, it can be the sequence of amino acids of the drug. And then I have a model, perhaps informed by AlphaFold and fine-tune on stuff of interest to me that maps this chemical structure into a prediction of how it's going to bind. Okay, and this I'm going to say that this model is independent of the training and test data we're going to discuss. Okay, now, okay. So I have some training data, right? So I have, pairs of fixed chemical features and measured affinity because, we're not the first to look at drugs. People have done it before. And so the training set is in green and blue. And so for all these drugs, I measure the affinity for the target molecule. And now the problem is I have a lot of test samples of the form xn plus j, yn plus j, with unknown labels. So I don't see the labels. This is the library I would like to screen to find the drugs that should be interesting to me with unknown values of the affinity. These are the new drugs. And so the goal that we're going to set for ourselves is to find large outcomes. So of course, I do not see the y's. This is important. I just see the x's. And so I want to see the y's that exceed some threshold. And so I want to find large outcome, yn plus j above cn plus j without too many errors. And so we're going to insist on having not too many errors because, I want to be able to give to Genentech, which is actually what we do. We want to give to Genentech a list of drugs that if they were to go eyeing out and study them, they would not waste more than a certain fraction of their time and budget. Now, this cn plus j can be set by users. It doesn't, you can set any way you like. It can also be random. For example, I want the new drugs that you're going to select for me to be better than some affinity that I've seen in the past. It can be also random. Okay, so these are things you can specify that can be random. Yes. It can depend on the type of drugs. You know, for example, you'd say, this is a very good question. By the way, don't interrupt. There's something wrong with my slides. I'm not sure exactly where this talk is going to go. But yes, the CN plus J can be drug dependent. So for example, the drug N plus J might be in a certain class of drugs. We have actually quite a few things of that class in the market. So to be interesting, it needs to be above a certain threshold. Here is another N plus J might be a drug in a part of chemical space where we don't have actually a lot of products. And so then you want to have to be lower, right? So absolutely. And so CN plus J is very interesting because it's quite, you know, you could use CN plus J to leverage diversity. Like you do not want only to find the same types of drugs. You want diversity and et cetera. Okay. Okay, so this is really what we're after. And I think what we're going to do is we're going to try to do this in a principled fashion, and which is a bit strange in this talk, is that this is going to be a talk about statistical inference, giving you like very tight type 1 error control guarantees without making any modeling assumption of any kind. And so that's a bit strange. So, all right, so we want to do that. And so my motivation is drug discovery, which is, again, we do with Genentech. You know, Google DeepMind has 400,000 new compounds for you to look at. So how are we going to make experiments? Like we can't. But even though I'm going to talk about drugs, you know, the same problem applies to, I don't know, suppose you want to recruit individuals, right? You have features of individuals. You have their X. This could be what you can glean from their CV, from blah, 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 from a lot of things. And Y is job performance. And you want to, before you bring people on campus to interview them, or candidates at a large company, you know, companies are doing this all the time. They get you a CV, nobody's looking at, you know, they embed it into X, and then you try to predict whether this is going to be a good employee or not. Well, how do you select employees that perform at a certain level? Yes, there was a question.
I was just curious on the previous slide about if the task is like very chemically diverse, or like affinity is also very molecularly diverse, or if you can train it.
The previous slide, in the CN plus J part?
Yeah, just the second bullet point of like physical chemical features. Like, so do you restrict it to any chemical groups, or is like idea just to have as much chemical diversity as you can fit in the box?
This is a secondary goal at the moment. What I'm trying to say is I can try to think about these problems as encoded into this threshold. What makes a drug interesting? And what makes a drug interesting might be that it's not like drugs I already know about. And so you can actually, you know, this is a new drug, you know, I'm in a completely different part of chemical space. Therefore, I'm going to use a C, which is a bit lower than if you want to get a drug that does something I already know what to do. But maybe you're still interested because maybe the inhibition rate of that drug is lower, blah, 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 that kind of stuff. Maybe did I answer your question correctly or not?
That's helpful, Frank.
What?
That's helpful, Frank.
Yeah. So you're pointing to something which is important, which I work with my student at the moment, which is what right now I'm not explicitly enforcing a diversity criterion, sorry. But in work we do now, we extend this work to explicitly enforce a diversity criterion. So we're trying to modify the work you're about to see to make sure that when you discover drugs, they're not all in the same location in chemical space. Yeah. But right now, we're not doing it. Okay. Any other questions? Okay. Okay, so I think this problem where you don't know the labels and you want to know whether they're above threshold, I think you see it in a lot of areas, not only of science, but also of marketing and recruitment and talent identification and all that stuff. So I'm just going to talk about drugs, but you know, it can be applied to many other things. So for example, universities receive so many applications that it's not inconceivable to think that some of it is going to be outsourced. And I'm interested in selecting applicants so that the graduation rate at UCLA is not 10%. It has to be above a certain level. It's going to be a very depressing campus if the graduation rate is 10%. Okay. All right. So we have predictions from our AI system, and we use things that have crazy names. MuHat. So we're talking about graph neural networks and stuff that I can hardly make sense of. So they make these predictions. So they predict the activities of these drugs. And so, of course, these are very noisy, and we would like to quantify the uncertainty in the predictions of my neural nets to understand and be able to prioritize what I should give to the biologist who is going to spend a significant amount of time and money into experiments. And as I said before, we don't want to use any model. I want to use the latest neural net model, and I don't want to, nothing is Gaussian. There's no parametric model anywhere. Okay, so this is kind of what we want to do. Okay, so, all right, so, if you look at Nature and other magazines, they say, well, the promise is there, but does it deliver? And it's not, right? So AI's potential to accelerate drug discovery needs a reality check. And this is because these neural nets are imprecise, they have a lot of uncertainty about their predictions, we follow them, but sometimes they lead us astray. And so it's not clear that we move in the right direction. And so what we want to do in this thing is how can we keep on using them, but so that we have statistical guarantees about what they recommend. And so can we draw discoveries with just a few mistakes? That's going to be the goal for today. So it was mentioned before, conformal prediction. Conformal prediction is a way of actually getting things about model-free alternate quantification. So conformal prediction is a method that you say, well, here's an xn plus 1, here's a drug. Can you predict the yn plus 1? And the affinity of this, and can you build a prediction set? So, typically the neural net will say, I think the affinity is 14.5. And what we've learned in the last 20 years, roughly, is that not only we know how to do this, but now we kind of know how to create a prediction set. That is, we know how to create an interval that depends on training data and past observation that would contain the true label, 14.5, a certain fraction of the time, right? Without any modern assumption, which is a great thing. So it's not a confidence interval because it's not about a parameter, it's just about a future observation. And so what we've learned, thanks to the work of Vladimir Vogt and his followers, is that, whether you have continuous or categorical outcome, there is technology that would transform a predictive algorithm into something that actually returns sets that are guaranteed to contain the true label a certain fraction of the time. And we're going to leverage some of this technology. So it's possible to use a prediction from neural net and transform it in such a way, you can think about this as a wrapper, that the prediction interval that you build for the n plus 1 drug contains the true affinity with probability 95%, no matter the prediction model, and we're going to leverage it, right? That's one way. We're going to not use any kind of, we're going to use a more fancy model because this works no matter the distribution of the data and no matter the predictive model you're using. Okay, so here's a challenge, I think, now is that, okay, so I have all my drugs, and you see that in some example, we might have millions of them, which is what happens with Genentech, with millions of compounds that we're looking at simultaneously. And for each of these, I know how to predict affinity in such a way that 95% of these rows, the true label is inside. Right? And so this quantifies uncertainty, and I make no model assumption. And so the question is, well, maybe I should get this guy. These guys, there's an interval for predicting affinity, which is large, and so I'm pretty confident that they are good drugs. Right? Maybe my cutoff is somewhere here. These intervals are right to the right. They look promising. So should I go and use them? So can I use them to find interesting instances of drugs? And this is what people are trying to do. They use conformal to do stuff like that. But as you can imagine, it's missing a key principle of statistical inference. there's going to be a very strong selection bias. Okay, so there's going to be a selection bias. And so here is a plot that shows the selection bias you're going to occur. So this is showing you on real data, actually, that conformal prediction works. So we took real drugs, and then we used these neural nets to predict their activity. We constructed prediction intervals, and over the library of drugs, for which we had labeled Y, which of course the algorithm had not seen, we set the miscoverage level alpha. So 0.1 means you're going to cover 90% of the time, the unknown label. And then we see that we have a straight line. That is, the theorem you saw is unbelievably, it's a theorem, like it's, okay. So you see a straight line. It's perfectly calibrated, perfectly. So this is showing that conformal prediction works. But now I could look at whether it works after selection. And now I'm going to go back to this picture, and I'm going to look at those intervals that are sitting on the right of my threshold. Do they contain their true label 90% of the time? And the answer is absolutely not. And in fact, you get the miscoverage, which is the orange curve, right? This is a phenomenon that is well known in statistics. There's a very strong selection bias, and therefore the properties of inference do not hold after selection. All right, so for example, this is absolutely nuts. You think you're taking a point which is here, you should have 99%, you should be okay. 99% of the time, this dot should be in here, but in fact, it's only 70% that it's in there. And if, of course, you're not going to select a lot, so if you go over there, most of what you're going to send to the lab is not going to pan out. Okay, and so this inspired this phenomena, which for parameters, not for observations, inspired a whole field of research called selective inference. And I don't know, I was very happy to see that earlier last year, the Russo Prize, a new prize in statistics, was awarded to Yoav Benyamini and Ruth Heller and Dani Yekutieli for fundamental contribution to selective inference. Okay, so what we want to do is, what we see is we want to scan through our drugs to find drugs, which are going to exceed the threshold, but I want to be able to control the fraction of false positives. Okay, so I should not call this the FDR, because the FDR is about parameters, but I'm going to abuse language anyway. I'm going to call this the FDR. I'm going to count my false discovery. So what is a false discovery? It's a drug I'm selecting. That is not, when I send it to the lab, I don't get what I want. Over the number of drugs, I want this ratio to be controlled. Like I want it to be 30%. I say, Emmanuel, you can afford some losses. It's okay to waste some of your money because we're going out, we're doing science. It's okay to follow false leads, but I want to control. I don't want us to follow 99% of false leads. And so this is a bit of a, I should not call the FDR, because an FDR is about a parameter. This is not about a parameter, but I'm counting errors, and I want that the fraction of errors be controlled. Okay, that's the FDR. And if I manage to find a method that controls the FDR, then it would imply that the AI-powered decisions are mostly correct, and that I do efficient resource allocation. So we're going to use our black box, and then we're going to try to control the false discovery rate, right? So in such a way that, okay, I think we know what that is. So we're going to do that, and I'm going to show you a method that does this, and it's quite simple. And then we're going to change it a bit to be able to deal with distribution shifts, which we always have in real life. So we're going to define hypotheses. Now, these are not hypotheses in the traditional sense, because a hypothesis is about a parameter of distribution. This is about the outcome of random variables. That's a bit different. So I don't know exactly how we call it, but I'm going to call it a random hypothesis that yn plus j, that's a null, that this drug is not interesting. And then I would like to construct a model free, because we don't want to make modeling assumption, to assess whether there is evidence that this molecule is not interesting or is interesting. So I want to construct a well-calibrated confidence measure which these are going to be conformal p-values essentially. Not quite, but almost. And so then we're going to determine how strong the confidence should be to be selected. So adjusting for multiplicity, obviously. So here's how we're going to do. So we're going to unroll this program. So first, we're going to construct what you should think of as p-values, even though we're not doing classical inference and we don't have a model. But I'm going to define a conformity score. And a conformity score is a function that takes as input the feature and the response, x and y. And I'm going to, for this talk, it's very important for us that this conformity score be monotone in y, in the second argument. So for example, it can be a y-sided residual. It can be the affinity minus a predicted affinity. It can be, following a bit more like Ryan's work, the affinity minus the predicted affinity over an estimate of the variability of my estimated affinity at that location in chemical space. Can be the CDF, or predicted CDF of the affinity condition on features. So this is what it can be. All these functions are monotone in Y, so we can use them safely. Now, I have some data, I have a training data, and so I can actually compute these scores on the sample data, on the training data, and I get a histogram, which is what we see over there. So we get the histogram of the VIs. And I cannot compute, of course, the test scores histogram because I don't have the Ys. So I'm going to compute test scores instead by replacing Y with a cutoff, Cn plus J. So I'm going to put in lieu of Y, the measured affinity that I cannot see, I'm going to put the cutoff instead, Cn plus J. And so this is, for the test graph, this is what it finds. You know, it's on the tail of the histogram. And then I'm going to compute a p-value. I'm going to say, if this were the affinity of your test molecule, then what would be its rank amongst the test scores that you could measure? And so we're going to look at the rank of the test molecule compared to the training molecules. And we're going to call this PJ because this is actually a p-value. We'll see this in a second in some sense. All right, so we're going to compute essentially the rank plus a bit of noise to make it continuous. So we're going to calculate the rank of these test scores with, instead of Y, imputed thresholds. Okay, so it's a rank among the training scores. And what you can think about this is you can think, we're going back to my diagrams before, you know, this is a confidence interval, a prediction interval for Yn plus J, for Yn plus J at level 90%, so 0.1% miscoverage. And what these p-values are is when you actually unroll the conformal prediction machine, is that you're going to say, okay, you know, this is to the right. And so what is the first level at which your prediction interval touches your threshold? And that's good. You can think about the p-values exactly in this sense. Okay, so for example, here it would say like the p-value is below 0.04 because the 0.04 is still to the right, and then maybe when it's 0.02, you touch the thing. It's the first time your prediction interval from conformal is actually touching the target you care about. Okay. All right. So we're gonna now set confidence about the IBH. We have different methods to actually now sift through these p-values and do selection. So we're gonna rank test these samples by p-values, and we're gonna apply Benjamini-Hochberg procedure. I have to issue an apology, which is it's a talk I gave in honor of Yoav, who got the Russo Prize, and I removed the definition of Benjamini-Hochberg, but I assume that it's okay for an audience. Everybody knows what the Benjamini-Hochberg procedure is? I think we do, right? Okay, good. So I'm going to apply the Benjamini-Hochberg procedure to these p-values. I could apply others, and we derive many other methods other than the Hochberg procedure, but we're going to take these p-values. Okay, so this is my Benjamini-Hochberg procedure. I don't think I need to dwell too much on this. And the surprising result is when you do something as simple as this, you're going to control the FDR, what I define to be the FDR. So if I have IID data, so, you know, I get a random sample from the population of molecules, and I apply this procedure, and then it is a theorem that this procedure will control the FDR at any level you like. And I think what's nice about this is that, you know, there's no reference to the prediction model, to the data distribution. The threshold can be random. And I think one thing that is very important to note for people, the people that are in the audience and know a lot about statistics, and I know a few of them, these p-values are dependent because they all depend on the training data. Like they all correlate. If the VIs go down, that it's an implication for all p-values simultaneously. So they are all correlated. Okay, so we have this stuff, and so yet no matter what, it's going to control the FDR. So so this is kind of a new type of statistical inference guarantees theory, which is like we are doing multiple testing, but for random hypothesis, where we're counting errors. And so one thing that makes everything work, that's only one piece, is that if I look at the event that the Y is not interesting, that's my random null. And if I look at what's the chance that Pj is less than T when sort of this thing is true, then it's less than T. So these p-values are, this number of Pj's, you should think of them as p-values. They're stochastically dominated by uniform. So this is what we get from these rank tests, so to speak. And then this p-value is a problem is now that they are extremely dependent, but they are positively dependent. And so they have a form of positive correlation, which is actually good for BH. And the one amazing fact about these p-values, which I'm not going to detail, is that these p-values are not PRDS, but they are p-values that are more conservative than p-values that are PRDS, and that's what makes everything work. Okay, so it's good for BH, and so we can apply BH. So now, how should you choose a score? You know, we can try to predict. There's many ways of scoring. So the score we're going to use the most is the score that looks like this, and this is a bit of a hack, but it's a score that we use. You know, we should use y minus mu hat, but if you use this score instead, you say, well, if y is above the cutoff, you're going to set the score to infinity. If it's below, you're going to use it c minus mu hat. It's actually, this score is, so we want to select, if you look at the procedure, it selects for small scores. And so this is going to push scores up. And so you're going to get more power by using a score like this. And that's the score we use in practice. So it's basically plus infinity if y is above the cutoff, and the cutoff minus the predicted value from your neural network if it's below. And so this is going to lead to strictly smaller p-values and therefore more discovery. Okay, so here's some real data now. So we're going to try to find active drugs for HIV with FDR control. And so, so here the response is binary. And so it's whether the drug interacts with the reverse transcriptase or not. There are 40,000 drugs in the database that we're looking at, and it's very imbalanced. Only 3% of these drugs are active. And what we want to find subsets with a certain percentage of active drugs. Okay, and so maybe we should look at the CLIP score. And so we see the realized FDR. So we do a 6-to-2 splits. So we see the realized FDR. And so I set my target to be 0.1, I get 0.09, 0.2. It's really, really spot on, exactly where you want it to be. It's not conservative. And then, of course, as you relax the false discovery rate, we get more and more power. You discuss more and more, you discover more things. And so this is where you tell us where you want to be. So for example, if we're willing to tolerate that half of experiments end up in failure, then we can explore 240 things. Okay. So this is what, and of course, if I did not do the correction for selection, then, you know, if I were naively to trust the prediction of conformal prediction, then the FDR would be through 95%. Of course, and that's a selection bias we've talked about a lot. Okay, so so here is another data set where we want to find highly binding pairs for drug target pairs. So this is again a large library, 30,000 drugs. Now the prediction, the target is continuous. And now kind of interesting, it's a bit what we're discussing before. Now the Cn plus J are random. They are quantiles of, so Cn plus J, so a drug belongs to a group. And the words is that what is the target of this drug? That's J, and then I'm going to look at historical data to actually select the sample. So it's the 70th percentile, the 80th percentile of the affinity of drugs I already know about. So this is actually random, but the theory says that's fine too. And so we see look at the red stuff. This is if I set my FDR to be 0.1, and so we see that it's okay, 0.2 is okay, 0.5. It's very boring plots, but for me, they're reassuring. Like you want 50%, you get 50%. You get 30%, you get 30%. You don't get 25, you don't get 53, you get 30. And this is a power. And of course, as I relax the FDR, I'm discovering more and more stuff. That's obvious. Okay. Okay, so this is when Ying went on the job market, I said, Ying, we live in the data science world, AI, you're going to have to show them like fancy neural nets and stuff. So, they are like neural IFE, CNN, graph convolution network. Morgan is something incredible. And so this is a form of external cross-validation where many things vary on these plots. What we have is we have different problems, and then we have different data, different neural nets to actually try. And of course, it's a theory, you've seen it already, but no matter the data, no matter the neural net you're going to use, you should get FDR control. And that's what you see. This is a 45-degree line. This is your nominal levels. This is the realized level. And you're always below. Okay. So that's good. Okay, so we know how to do this. The reality is that life is a bit more complicated than this. Okay. In reality, what happens in most problems is that we have the training drugs on which we know the affinity, and then we have the new drugs. And it's not true that they are IID sample from a distribution. There's a simple reason for that, is you are in the training data because you look interested in the first place. And so there's what people call a distribution shape. Now, the functionality or the shape or whatever you want, given the chemistry, it doesn't change. But the distribution of X changes. The distribution of Y given X, it's chemistry, it doesn't change. But the distribution of X changes. So, in general, you have in your training data things that are interesting, and in your library, things that are perhaps less interesting. So you have a shift. And in reality, that's what we see. And so we're going to need to deal with that. And so what's interesting in this problem in drug discovery is that we only have a coverage shift because we have, like, I don't know how you call it, Schrodinger's equation does the rest. Right? Once you give me chemical structure, it doesn't matter whether you're in the training set. You're going to fall the same way. Doesn't depend on whether I put you in the training or not. And so what matters is when we look at the likelihood ratio between the distribution of the test data and the training data, it only depends on X. Okay, and so, and this is also true in a lot of examples I showed you before, which is that if I hire people, I only have the Y for the people I actually hired, for the UCLA students I admitted. And so you have a covariance shift. Okay, so but we still want to do this. And so now what we're going to do is when we compute our rank test, we are going to be more careful because now there are data points in the training set that we want to upweight and data point that we want to downweight. So intuitively, if there's a shift between the training and the test set, I want to give more weights to the people that look like from the training set that could have come from the test set, and downweight those are very unlikely under the test set. And so maybe since my time is almost up, I'm just going to say that the way you do this is very simple. When you do your rank comparison, you weight the scores by how how likely are they to come from the test set? And so the only difference is that you're going to add weights. So that's at the level of the methodology. So you compute a p-value where you're going to replace, this used to be 1, but now it's going to be the likelihood ratio. Okay, so that's what you do. And so it's a weighted rank now of the test data among the training scores in such a way that you adjust for the distribution shift. And this, if you do this the right way, these measures are still valid statistical measures. That is, these are still p-values. They're still dominated by the stochastic, the uniform. The problem is that they're not PRDS. Because now when things move, it's because, you know, there's these weights, and so there are no PRDS. They're no longer, so you cannot use Benjamini-Hochberg. And so the weighted conformal p-values, for important reasons, are no longer positively dependent. It's not true that these p-values are positively correlated. And so now we can't use BH. So what we had to do is we had to invent new method. When I say we, I'm generous. Ying had to invent new methods to actually get out of this problem. And so we constructed new selection methods, which are not unlike BH, but are quite different. They're both more based on e-value theory for people who know about e-values that would actually deal with this form of dependence so that we still control FDR. And so this is what we wrote about. And so we have a method, we have new methods that would actually sift through these p-values and will control the FDR. Okay, and so I'm not discussing what this method is doing, but it's related to e-values. Okay, so here is another example where we have a very clear distribution shift. This is the predicted activity score in the training set, and this is in the test set. You see that in the training set is higher, because that's why you measure this, because their predicted score was higher. So it's higher. And so when we apply this new method to, which is not BH, we also applied BH, by the way. BH seems to be fine, even though the theory says it's not fine, it's fine. So this is the blue box plot we control, and this new method, the yellow, green, and purple provably control the FDR. As you can see, they sort of behave similarly to BH, and they are provably controlling the FDR. Okay, so maybe, is it time to wrap up? 1 minute. So as I said before, we are working with Genentech on this. And so Genentech has stuck in this, and with us, with our help, obviously, to actually do real drug discovery in the wild. And so here you're looking at real stuff that we're experimenting with in the lab. And so we're trying to select drugs with different properties, and maybe I'm not going to go through all these plots. They're very diverse. One is about trying to find drugs with low inhibition rate. One is to try to find proteins or drugs with which are very stable, that they will not degrade easily, and so on and so forth. One is actually to predict the outcome of clinical trials, which you can also do, which is example number 4. So this is selecting protein with high biopolarity. This is selecting genes perturbation, which will lead to a high T cell proliferation. And in all these plots, they're very boring. This is a method if you use conformal without adjusting for selection. And so you have huge problems. You have enormous violation of FDR. And this is if you use our method. They all have distribution shift except number 1. And so you see that these are very boring plots. You want to hug the 45-degree line, and this is what you do. And you'd say, why does it stop? Well, it's because there are no more discoveries to make. Now you have exhausted the thing. You have made all the discoveries you could, and there's nothing else to actually find. And so no matter the setting, when we apply this method, we see that we're hugging until there's no more discoveries to be made, and of course. And these are very, very different scenarios with enormous distribution shifts, and the method seems to work quite well. Okay, so we can deal with distribution shift. I'm excited about this because I love first discovery rate theory and this stuff. I also love conformal methods, the model-free things, and now this is trying to put them together And so, yeah. And Clara says this, so it means I have to stop.
We won't have time already. Oh, okay, sounds good. Yeah, we will have one time.
So when you were starting out and saying, we really don't want to do any modeling, I didn't understand.
No, we do in the prediction model.
Until you were trying to tie it in with the false discovery.
Right.
So, well, that makes sense, but I guess then what I don't understand would be, once you have some discoveries that meet the false discovery rate criteria, why wouldn't you want to use modeling strategies or something maybe to prioritize the, combine that with another procedure or something? I guess it seems like you were sort of going off in a different direction of like, well, if there's some other modeling, or some other selective process, then let's come up with a different procedure to maintain the rate. Why not first do the false discovery rate and then prioritize based on model? It's sort of an overarching question.
So first of all, you have the right to make models. This will actually, I could use your modeling strategies and put it into my conformity score. Whatever you think is great, I can incorporate everything you know about the problem, every model you want. And that's a bit the freedom I've not talked about, which is that I can put this, right, I can put this in my conformity scores, like here, which is actually, I'm going to use this. So any model you want to build, I can use. When I say I don't use a model, I mean that the joint distribution of X and Y, it is arbitrary. I'm not going to say, oh, if Y is 0, 1, it's binding or not. I'm not going to say it's a logistic model, and I have to believe it so that the properties of inference that I'm going to lay out actually depend on this. And if it's not a logistic model, then you're on your own, right? No, this makes no assumption about how Y depends on X and how X, the X covariates depend, relate to each other. That's what I mean by no model. But of course, in our work, it's not models. I don't think we can then call models in the statistical sense, but in the V, we're going to put a lot of stuff we know, right? So we're going to try to use the best neural nets, the best embeddings that we can find to, of course, pump up the true positive rate.
Thanks, Dr.